Hello everyone. In today's video solution for life sciences gate question, I'll be mainly talking about the protein overexpression in the different host. Here, protein expression and purification topic under the topic. This question I have selected, which has come in the life sciences gate question paper in 2016 in biochemistry. This is question number 19. So, this question states that an N-terminal histidine tagged protein of molecular weight 40 kilodalton was purified using nickel NTA column. This protein sample was subjected to STS page. A Western blot of the same using anti-His antibodies is shown below. So this is the Western blot, which is showing two bands, one at 40 kilodalton, another at 20 kilodalton. So which one of the following interpretation is correct? First, it says only the histidine tag of the protein got removed. Then the protein forms oligomer. Third is the purified protein sample is homogeneous. D, the protein has a stable N-terminal 20 kilodalton domain. Okay, let's talk about this question. So firstly, it says N-terminal histidine tagged protein of molecular weight 40 kilodalton was purified using nickel NTA column. So this protein, so generally what is we do, we perform many heterologous expression of proteins. So suppose if I'm working on any human protein and I need that protein to perform several assay, so what I do, I put the gene sequence of that protein, okay, inside the bacteria. So this is a bacterial cell, okay. So here inside this particular bacteria, I put the gene of another, my desired protein, my gene of interest, okay. So inside the bacteria, this protein will overexpress. So what we do, we tag the so here they have tagged the N-terminal of the protein with histidine, okay. So here one tag is present in the protein, that is histidine tag. So whenever this protein is being expressed, so this also carries a N-terminal tag, okay, which is the histidine tag, okay. And using this tag, the protein purification can be achieved, okay. So the cell being lysed and suppose different proteins come out, many different proteins, because bacteria has their own protein with their own machinery, and in between them this histidine tag protein and terminal histidine tag protein is also being expressed, okay, other proteins, okay. So after breaking open the cell, many proteins will come out. So what we are using nickel NTA column or nickel NTA beads. So these nickel NTA beads have a tendency to bind to this histidine tag, okay. This histidine tag will be bound to this beads, okay, because all other protein does not of the bacteria does not contain that tag. So that tag the beads will not bind to the other proteins. So in our purification, what we'll have, we'll be able to separate only the histidine tag protein. So this is the histidine tag protein, because it was bound to the nickel NTA beads. So we are able to separate that from other proteins. So other proteins will be removed, non-specific protein which does not have the tag, and our desired protein will be purified, okay. Our desired protein will be purified using this nickel NTA column. So what they have done after the purification step, they have performed a Western blot analysis. That means this purified protein they have loaded on a STS page. They have loaded this protein on STS page and they have performed Western blot analysis. In Western blot, what we do, we transfer the protein from STS page or polyacrylamide gel to the nitrocellulose membrane. Okay, so you transfer the protein on a membrane. So if you have purified this protein and they are saying 40 kilodalton protein, that means your purified protein is being transferred here. Okay, that will be only visible. So here you run a molecular marker. Okay, so that being transferred. So this protein can only be visible using Western blot when, okay, so after proteins are being transferred, you probe them with antibodies. So this antibody that they have used is against histidine tag. Okay, so that is why it is called anti-His antibody. So this anti-His antibody they have used. That means this antibody. So this is the histidine tag. This is the protein. So this antibody will bind to this histidine tag. Okay, will bind to this histidine tag, and another antibody which is so this is the anti-His primary antibody. Another antibody which is being used is the secondary antibody, which can recognize this primary antibody, and this secondary antibody is usually is conjugated with the chemical reagent enzyme HRP, okay, horse radish peroxidase, which upon giving its substrate luminol and hydrogen peroxide will perform the reaction and give us a band that can be detected on the membrane. Okay, so as you have loaded a purified protein of 40 kilodalton, when you are performing the uh, um, final developing the blot according to molecular weight, if you have 40 kilodalton protein purified, you should observe one band that corresponds to 40 kilodalton, and that band should be visible. Okay, 40 kilodalton band upon the developing the blot. So here what is happening, instead not only 140 kilodalton band, they are getting another band that corresponds to 20 kilodalton. Okay, so they are getting two bands. Instead of getting one band which should correspond to 40 kilodalton, they are getting one more band that is corresponding to 20 kilodalton. So that is why they are asking what is the reason of getting this extra band. So in order to interpret this result, they have given this four options. So let's consider the options one by one. So first option, they are saying the only the histidine tag of the protein got removed. So they are saying that this histidine tag is being removed. Okay, and protein is being separated. So if the tag is being removed, and this antibody is which is used again this tag only, then only the whole protein can be recognized. Okay, so if the tag is removed, the protein cannot be detected. Okay, if the tag is removed, very this entire 40 kilodalton protein will not be detected with the antibodies. So this band will not be visible on the western blot. So 40 kilodalton band will also not be visible, but still we are able to see the 40 kilodalton band. That means the tag is still present. Okay, or if the tag is being removed, no band will be visible because the antibodies which you are using, that is against the tag, that is anti-histidine antibodies. So if the tag is not there, bands will not be visible, but still you are able to see the bands. That means the tag is intact. So first option is wrong. Second option states that the protein forms oligomers. Okay, so protein form oligomers, they are not saying that they are being removing the tag. Okay, that means tag is intact. Okay, so tag is intact and this protein forms oligomer. Oligomer means more than one copy, between 2 to 10 copies of the protein. So many subunits of the protein, or many the protein is in the many different units are being joined together and forming a protein. Okay, so because many different subunits are being joined, that is why when you are running it on the SDS page, those bonds will be broken and subunits will be divided. So that is why you will be able to see different bands of, okay, more than one bands. But if the protein actual monomer size, okay, one is monomer if it is forming the oligomer, so the monomer size is 40 kilodalton. Okay, if it is forming the oligomer, okay, if it is forming the oligomer, you should see the, if two proteins are being joined, if two subunits of protein are being joined, that means it is forming a dimer, then 40 kilodalton two proteins will be associated together. That means the size should be come further above, that means 80 kilodalton you should see a band. Or if three protein units are being joined together, that means further above 120 kilodalton. So that means in any case, if it is forming an oligomer, the protein, if it is forming an oligomer, the protein or the band, corresponding band will be higher than 40 kilodalton, not lower than 40 kilodalton. But in this case we are getting a band of 20 kilodalton, which is lower than 40 kilodalton. That says that no, it is not forming oligomers, because if it is forming oligomers, the sizes will be above 40 kilodalton. So that is also not happening. Okay, so the second option is also wrong. The protein is not forming oligomers. Third option is the purified protein sample is homogeneous. So as you are able to see two bands here, that means it is not at all homogeneous. Homogeneous means all of same kind. But if here two different kinds of bands only you are able to see, 40 and 20 kilodalton, that means it is not homogeneous, it is a heterogeneous purified sample. Okay, so the third option is also wrong. So fourth option, that says that protein has a stable and terminal 20 kilodalton domain. So the protein has a stable and terminal kilodalton domain. So that states, that means, like if this is my histidine tag, this is my 40 kilodalton protein. Okay, so if it has, so this is the N terminal, this is the C terminal. So it has, if half of the protein from N terminals, okay, that is being produced, and that is a stable protein, forming a stable protein. Okay, and because it is an N-terminal domain, it will also contain the N-terminal histidine tag. Okay, so now both of the proteins are being expressed and both of the proteins are stable. That is why both contain the histidine tag also. That is why when you are performing the Western blot using the antibody, because of the presence of N-terminal tag, both of the proteins are being detected. That is why you are able to see two different bands, one corresponding to 40 kilodalton, another corresponding to 20 kilodalton. That means fourth option, the protein has stable and terminal 20 kilodalton, is the correct option here. Okay, so even though 40 kilodalton protein is being produced, we are able to see another size, another protein which is of the size of 20 kilodalton. That means this protein also contains the histidine tag, and it could be the N-terminal 20 kilodalton domain of the protein. So the fourth option is correct. This is the NPTEL reference where you can study more about the eukaryotic gene expression and purification. Thank you.